he uses mathematical models to collect other phenomic information about roots. And so as we have multiple time points throughout growth, and we have multiple, things we can extract from images, we can start getting a handle on the phenome, which would be holistic, just like the genome is measuring, hundreds of thousands or millions of markers across the genome is genomics, versus genotypic is usually, a single cultivar tracking a few, important markers, rather than, whole genome sequencing or something like that. I hope that helps.
Yes. We have more questions, but I'm just going to read one last question from Gideon. Dr. Murray, could you adjust your video? We're live streaming on Facebook and your video is kind of, yeah. Thank you.
I don't know how my video was before. Is that better?
Yes, it is
okay.
Better. Thank you. There is one question from Gideon. He is from northern Nigeria and he works on a cowpea breeding program. And he's wondering if the UAVs can be deployed to phenotype for striga and electra resistance. Could you give a rough estimate of the level of precision that could be obtained?
Wow, that's a very tough question. Striga and what was the other one?
Electra. They're very similar. They're okay. Parasitic weeds.
Yeah. No, I know striga. I haven't heard of the electra.
They're like very close relatives.
Okay. So I would imagine, I haven't done this and I haven't seen anybody do this, but if you do it, you'd probably be the first to do so, and it would be a very novel and highly influential publication, I'm sure. But if you were to fly over a field with striga, I would think, since striga is white, as I recall.
It's green.
It's green. Oh, okay.
Purple flowers.
Okay. Well, I was thinking it was white, because if it was white, then the vegetation indices would, you'd be able to play with your colors to really highlight it and make it pop out. But if it's the same color as your plants, that would make it a little more difficult. But through approaches, either by hand, you could look at each plot and count the amount of striga, or measure the number of pixels in it that have striga, but that would be very labor intensive. I could also imagine approaches with machine learning where you might train a data set to capture the striga, and then you could run that on future images. I don't see any reason why it wouldn't work, except if the striga is underneath the plant canopy.
Yeah, right? So yeah. That's what I wanted to say. It's under.
Right. In maize, the ears, for instance, we can't measure ear length or ear height because it's under the canopy. But I believe we can correlate some of these traits to ear height, right? And also, if the striga is negatively impacting the plant health, the vegetation indices on the maize might be able to help you see that. Finally, when those maize plants dry down, if the striga is still alive, they should stand out, very strongly. As the leaves fall down, depending on the type of maize you have, if the leaves fall down and you can see the green plants underneath, that may be an indication for you too. I hope that helps.
Yes, I think that is great, because since striga affect the vegetation, and striga is actually hidden under the canopy of cowpeas, you have to, move the cover in order to count the number of striga. I think the vegetation and the impact on the crop can be used to correlate. Yeah, that's okay. Great. Yeah. So
I'll continue to monitor the chat, but I'm going to turn off my video to save battery, and our power's been out for the last 45 minutes. So I need to check on some things. So I'll pay attention and answer the chat. And thank you.
Thank you so much, Dr. Murray. We appreciate you joining us today. Thank you. All righty. Yeah, so the next person we'd like to hear from will be Alper, because Alper's network is also not um, really good today because of the weather. So I will briefly introduce Alper. Alper is a PhD candidate with Dr. Seth Murray, and in the quantitative genetics and plant breeding program in corn. And his research interests include high throughput phenotyping, statistical analysis of temporal data, and molecular quantitative genetic discovery. So there is a question here in the chat about some temporal data and disease curve. So his tutorial covered ggplot, details on ggplot. He did an exhaustive video on how to optimize ggplot. He also did some analysis on high throughput phenotyping data, like the flights you receive from your drone images. If you are working on high throughput data, I will encourage you to follow his tutorial step by step. And he also covered correlation. So I will let Alper do a recap of his tutorial. Alper, over to you.
Thank you, Catherine. So I'm sorry for my internet access because Texas is not like ready for that kind of harsh weather. So unfortunately, every pipelines and the cable system are broken in here in Texas, which is really dramatic and funny though. So yeah, personally, as a PhD student in quantitative genetics and maze breeding in Texas A&M University, my interest is to combine high throughput phenotyping with high throughput genotyping to understand especially the phenotypic plasticity of complex traits such as plant height, because plant height is one of the easiest traits that we can extract from the drone images regularly. And there are like recently published many publications of many crops, and especially maize, sorghum, and there are many other examples. I'm sure you are familiar with all of them, but when it comes to illustration of the temporal data, which might be a little bit tricky, so I spent many time for that, and I recorded a video that like around 1 hour long, but I used my data, but I'm sure you have a like spatial data structure, and if you have like any spatial problem, let's say, I will be happy to help you if you send an email to me, and I can like share my experience with you if that helps to you. I'm sure many of you are familiar with ggplot2, which is one of the like extensively used package in R. I also use that, but sometimes I like think about that we are flying drones like 20 times, 60 times, and then we are extracting like 15 to 20 different vegetative indices as well as plant height. So when we multiply the flight times with the traits, that creates an enormous big data. And so we have to present that somehow. So I put one of the example in my recording so you can see that. And also, I saw one of the question the person is asking area under curve, for instance. This is one of the common application that can be easily calculated for each trait once you have the data for, once you have the temporal data for specific trait, for instance, and you can calculate the area under curve. Area under curve is just one of the approach that you can calculate for, especially for disease. But when it comes to plant height or the various vegetative indices, you can calculate the same area under curve parameter, and you can differentiate your genotype of interest based on this knowledge. But unfortunately, so far, another point is that in the temporal analysis, unfortunately, many people so far just evaluate every time point, every flight time as a single time point. So that disregard the hierarchy of the plasticity of the phenotypic plasticity of any complex trait. So I also would like to offer to you to use such as nested design that has not been used before. So that might be one of my interests nowadays. I'm using the nested design to illustrate the phenotypic plasticity of any vegetative indices or the plant height in corn, and it can be applied for any other crops, for instance. So there are many examples, but in this session, mostly I focus on the illustration of the vegetative indices and the plant height. So I will be happy if you have any special question to deal with your data, a special illustration of your data. So with this, I would like to conclude my recap, and I also shared my email address in the chat. So I also am going to follow the chat if you have a question to me. So that's all. If you have any question, I will be happy to answer.
That's great. That's awesome. Thank you so much, Alper, for taking the time out to prepare the tutorial. So if you are really new to R and you have not used ggplot before, I encourage you to watch the tutorial from Alper. He did an exhaustive video on ggplot, which is one of the most used R packages for basic visualizations of your data. And he also covered some aspect of high throughput phenotyping and correlation. So if you're stuck in your code, we expect that you learn how to do the analysis, but if you're stuck somewhere while analyzing your data, feel free to reach out to Alper, and please copy or CC the JR Biotek Foundation in your email so that we keep track of the communications that is ongoing. So I will leave the chat open for anyone who has questions based off of the tutorial from Alper. And we will be happy to take those questions as we move ahead with the remaining parts of this workshop. So I don't know how many of you watched the video from Alper. Can you indicate, some of you just indicate in the chat. Did anyone watch the tutorial video from Alper? Okay, all right. Okay, that's great. So I encourage you because, oh, awesome. So I encourage you to, the R script was provided. The data set he used to demonstrate was also provided. So the only way you learn how to do this is by doing it yourself. Since you have the data, you have the R script, I encourage you to keep practicing with your own data. I'm sure you all have data with, you know, your field data or greenhouse experiments. It is really important that you practice because we have a poster competition coming soon, and we would like to see those pretty plots showing the summary of your data. So thank you so much for that quick recap, Alper. And if you have questions for Alper, you can type it in the chat, and we'll see how we can answer your questions. So moving ahead in the workshop, we have our next speaker. Our next speaker is Jales Fonseca. Jales was born in Brazil, and he is a PhD candidate at Texas A&M University under the sorghum breeding and genetics program in Dr. Rooney's lab. And his project is a collaborative effort between Texas A&M University and Kansas State. And he is accessing the power of genomic prediction in sorghum breeding programs to predict hybrid combinations based on inbred lines and genomic information. So for everyone who is working with corn or sorghum or some kind of Diallele crossing, Jales has the expertise and the statistical capacity to answer your questions on how to easily analyze your data on, you know, inbred lines, SCA, GCAs, and all of those things. So we really appreciate and thank Jales for taking the time out to prepare the tutorial video. It's on the JR Biotek YouTube channel, so feel free to watch it if you haven't. And I will open the floor to Jales. Thank you, Jales.
Oh, it's my pleasure. Thank you very much, Catherine. Thank you for the opportunity. And thank you, everybody that is attending. It is a great pleasure to be part of this initiative. And as you mentioned, here at Texas A&M, I'm working in sorghum, grain sorghum. And my project is basically trying to develop a genomic prediction model to predict hybrids based on the genomic information from the inbred parents. And because we're using the inbred parents, we need to set up crosses. And from those crosses, we can extract GCAs and SCAs, as you said. So especially for sorghum or corn, crops that are, they have their heterotic groups well established. We have our female side, or the female and the male side, or the restore lines and the maintainers and the A lines. We can develop some dialleles. For example, the North Carolina 2 is a factorial mating scheme. And with that, we can extract GCAs, which are the information from the inbred parent in a hybrid combination. So you can measure the value of a line in a hybrid combination. And also we can extract the SCAs, and with all these information, we can predict hybrids. So this is one of the applications of developing diallele crosses. And just a brief review here, just want to point out, I would like to share the screen. Yeah, I don't know if you guys can see it, but this is pretty much what my talk covered. Can you see this?
Yes, we can see.
Yeah, just a very brief review, because when I was talking about the diallele, any analysis of diallele, I wasn't put any focus on the theoretical part of it. So I was not trying to cover all the aspects of selecting an F2 or measuring the covariance between relatives. And my idea was literally to just define what is a diallele, which is just a set of crosses. And between genotypes, it can be used in plants, it can be used in animals. And those informations are really important for you if you're, for example, starting your breeding program. If you don't know what inbred parent you should cross to start a breeding population. So you set up this mating scheme, and you start crossing your inbred parents, and then through the statistical analysis of those mating schemes, you can see the value of that line in a hybrid combination. So you can see what parents you should use to increase the performance on the following generation. So you can use GCAs to create breeding populations and extract future improved inbred parents. Or you can just try to see what hybrid you should select, and then start making those multi-trial environments where you see the stability across, sorry, can you still hear me?
Yes, we can hear you.
Oh, sorry. Somebody called me here, sorry. So you can either find the best parents for you to cross and create breeding populations to develop improved lines for in future generations, or you can use those diallele design to find hybrid combinations, and then for you to determine which hybrid you should release, you need to create those mega environmental trials and see the stability and all that. So for you to do those analysis, and you need to set up those crosses, and based on how you set up those crosses, you can either focusing on a full diallele where you cross everybody with everybody, or half diallele, partial diallele. And once you establish that is, so you establish that at the very beginning of your project. So how you're gonna, so for example, if you have a hybrid crop, you will set up the females with the males, and then you will cross, or maybe you want to just identify the best inbred lines from one side of the heterotic group. So you can make a Tess Cross, which is, so you're going to get a tester from the opposite heterotic group, and then you're going to cross with, let's say, if you want to see which female is performing better in hybrid combination, you're going to select a lot of females, and you will have a tester that is a male, 2, 3 testers, and you make a line by tester mating scheme. And then you can classify which inbred line is performing better based on the GCA estimates of each inbred line. But before you do all that, you need to see if you have enough variation within that pool. And for you to assess that, you need to calculate the GCA, the variance of the GCAs of those lines. And for you to calculate those things, there are several models out there. You can use ANOVA, which is a little bit outdated, because they require very specific formulas, and very, you need to have your data set balanced. And we know sometimes we just lose a lot of plots. So we have other more flexible models, which includes linear models that use ordinary least squares estimators, or even more flexible models like the linear mixed models or Bayesian regression models. And that was pretty much what I was talking about during the lecture. And I was just giving some examples. And I hope you guys saw that. And I think with that, I could take questions. And it is a pleasure. My email is displayed here. So I think with that, I finish my recap, because otherwise I'll be rephrasing myself from the lecture. So I don't know, maybe it would be more beneficial to the attendees to start addressing questions. So thank you for your time, and let me stop sharing this here.
Thank you so much, Jales. In fact, I was watching your video last night, and I realized that you need to watch Jales's video at least twice. Because, all of the expected mean squares, he's just going through all the formulas and, all the complications of Diallele crosses. So yes, Jales did a great job showing us the different ways to analyze Diallele crosses. I know that Diallele crosses is what a lot of plant breeders in Africa do for corn, which is maize and sorghum. So it is a very great tutorial that Jales has provided for the African community. You can just input your data and just follow the steps, and you would analyze your data yourself. You don't need to pay anybody to analyze it for you. You can follow Jales's tutorial, save your money, and even teach others. When you learn, save your money, follow the tutorial, analyze your data, be confident, because Jales did a great job in the tutorial session. So thank you so much, Jales.
One thing that I would like to point out is sometimes it is a little bit scared, like it scares us to look at all of those formulas, and it's a little bit challenging to keep track of what R is doing. So I just want to make you guys feel comfortable of the frustration you will get in the beginning, because it is time consuming. It requires us to do some sort of try and error. So you will see, even with my my codes, you will experience maybe a problem with your data, like missing data. And so it will, you will find some challenges still with the code. But using that script, I think you will have a good idea of what's going on. The first part of my script, I literally teach you guys what was proposed in 1956 by Griffin. So all the formulas and everything that those incredible scientists developed in like half a century ago. So they were using ANOVA type of analysis. And in those type of analysis, you have a well-structured way to get to your the value of the significance of your effects. And it is important to understand that if you have fixed effects, you need to test your effects with different effects. So make sure that some of those concepts are clear, in a sense that, okay, so this population will be the population that I'll be selecting from. If this is the population you're selecting from, you're calling probably that population as fixed, and all of the estimates will be derived from that population. But if you're trying to expand those inferences for the host, for other populations or other individuals on your populations, you will call those effects as random. And there are some differences in how you're gonna assess the significance of those effects. But I think I went over in a little bit more details, and don't be intimidated by the formulas. The formulas are there. Somebody already developed the formulas. We just need to understand how to use it. And there are a lot of brilliant scientists out there and great coders, data analysts that developed this extremely useful packages. And we just need to know how to use those packages. So be confident. My email is there. Just reach out to me. One thing that I would like to point out is I got an email earlier this morning saying that they were having a problem with the plant breeding package. So I want you guys to see, can I share a little?
Yeah, you can share your R and show how to,
Yeah, so I can send an updated version of the script, but because this plant breeding is not, it's kind of outdated, you need to download from the repository.
Yeah.
So this is the line of codes that you need to put instead of just install.packages. Oh yeah, I can post this
in the chat. Yeah.
Yeah, I can just copy and paste this. And this is the code that you should put instead of, it's too old. Okay. Yeah, so I see that somebody, is already asking me about it.
There are so many questions already for you.
I'm sorry, I think I'm taking too much of your time. So make sure that for plant breeding, you need to download the package from the repository. So you just need to add some to your code and it will work. But I think, yeah, now I'm okay with that. I will take any questions. Thank you.
Okay. Thank you. So one thing I realize is I think we need to do a detailed lecture on Diallele crosses to cover the basics for those who are not able to follow the basics of Diallele crossing. I think we need to do a separate lecture to cover the basics. What is Diallele? I know you kind of did an overview, but your focus was on the analysis. So that's really great. So I'm gonna read out the first question for you here from Tolera. Is it possible to analyze GCA and SEA from unbalanced crosses? that doesn't follow the Diallele fashion.
Yes, yes. So that's what I was saying. For example, back in the days when you need to just use ANOVAs, a more closed type of analysis, you would be a little bit more penalized with your estimates, wouldn't be very robust if you have unbalanced data. But nowadays we have linear mixed models.
Yeah.
So if you approach linear mixed models, you will get a more flexible analysis. So your model will be more flexible, so you can account for that unbalanced data. So in your case, say you can analyze GCAs and SEAs. You can analyze SEAs if you have a good amount of hybrid combinations. But if you have, for example, a line by tester and you have only one tester, your SEA's estimates will be confound with the GCAs because you don't have enough hybrids for you to actually see the specific combinations that will give you some sort of different values across the mean performance, because the GCA, again, is the value of that inbred line over several different crosses. So it is the mean of the inbred line. But the SEA is whatever deviation from that mean due to a specific hybrid combination. So you will need to have more than one tester, than two testers. So the literature sometimes points out how many crosses should you have, but I mean, be confident that it is possible that you can analyze your data with unbalanced. You can analyze your trial with unbalanced data, no problem. Once you have your linear mixed models adopted and understand the dynamics behind of those models, because it is a different concept. Instead of testing the significance with a NAF test, you're not going to use a NAF statistic test to see the significance of your values, of your effects. You will probably use the likelihood ratio test, which is kind of a deviance test where you have the full model and then the reduced model where you take out the effect of the reduced model, and then you compare which one is, if you can explain. So if you can explain a model without the effect, so that effect is not significant, and then you use 1 degree of freedom in a chi-squared distribution. Too much information, but all the packages are there. Follow the structure. Don't be intimidated. It's all there.
Yeah.
But yes.
So, yeah, that's
yeah,
great.
Short. Long story short, yes, you can.
Thanks, Jales. So with newer R packages people are developing these days, it is very possible to analyze unbalanced design and get your GCA and SEA effect. It's just that it may not be as straightforward. You have to study the model and know how to extract the information that you need. So be sure to follow through with the tutorial, and then if you have questions, then you can email Jales. We want you to analyze your data by yourself. So we don't want you sending your data to us to analyze. The goal is for us to show you how to do it so you could do it independently. So we have another question here from Vincent. His question is, does the tester stand for the male parent in the crossing?
Stand for the male parent.
Is the tester the male parent?
Well, it depends. So if you're testing females, the males will be the testers. So in sorghum, in corn, for example, if you have a non-stiff stalk and a stiff stalk heterotic groups, if you're testing the stiff stalk, the testers will be a non-stiff stalker, a non-stiff stalk. Well, probably in Europe, you will have a flint heterotic group and a dent heterotic group. So if you are trying to see the GCAs of the Flint heterotic group, individuals from the opposite heterotic group will be used as testers. So the answer is the male can be the tester if you're assessing the females.
Yes, that's great answer. So there is someone here who is wondering how to include blocks, environment, location, and year in the R program for the Diallele. That's from Abdul.
Oh, where is it?
Yeah, Abdul, he wants to be able to add like blocks, environment, location, and year. And he's wondering where that would go in the R script.
Okay, so if you look at the
could you share your R?
Oh yeah, well, I can. Yeah. Sorry, I was thinking here, and I have everything here. Yes. So can you guys see this?
Yes.
Is it clear? Is it not too small?
Clear. No, it's. You
All
can
right.
zoom in a little bit.
Yeah, I can zoom in here.
Yeah, I think that works.
Is it enough?
Yeah, that's good.
All right. So let me see. When we start talking about the more flexible models, I'm little, it's too much here. Okay. So you can include using the what is the name of that package? The LM Diallele, and if you look at the VigNet, you can include. So here, for example, they put block, and block will be the rep.
Yeah.
So you can there's another discussion on that regard. So sometimes people analyze the data using one step, where you include reps, location, or even years, and you have everything in one model. So you will have a one-step model, or you can have a two-step model. And the two-step model means that you're gonna have your means across environments. So for example, you have 3 environments, and you have a observation in plot 1 for location 1, plot 1 for location 2. So you will have a mean, and then you will, excuse me, you will analyze your data on the means and not on the raw data. And that's a two-step. But answering your question here is a way for you to include environment within the blocks within the environment. So there is a way for you to include more effects in your model. If you have a factorial type of mating scheme, I would recommend you to use let me go back here. I would recommend you to use this package here. Here, this one. LME4, because it's very fast. It's very fast. I showed you Sommer because Sommer have a very good function called overlay. So if you have a diallele where the individual operates as a male and a female, you don't need to specify the matrix to do the analysis. So this will do it for you. But there is a way. So and it's pretty simple. You just add reps, rep effect, block, year, location. There is a way for you. And I would strongly recommend you to use linear mixed models for that. Hello? I can't hear you. You're muted.
I can hear you. Can you hear me? Catherine, I can hear you.
Yeah, I can't hear you, Catherine.
Yes, we can hear you. You're on mute. Okay, I think we have to unmute you.
Yes, I'm back.
So,
So, yes.
I think
Can
I
you
can.
hear me?
Yes, now I can. I will. I have a code here, and I will add this code on the chat as a way for them to
Um,
put.
yeah, so I'm thinking, Jales, that maybe you can update a section of your R script to include the unbalanced design and the, just to show like how to include like the environment like you showed and just kind of
Yeah.
update that
Well,
region. Yeah.
it is
Yeah.
there. So
Yeah,
if you
it
go
is there,
over,
right?
it is already there. It is
Yeah.
already
So
there. The
you
only
just need
reason
to
I'm
study it. Yeah.
it is there. So there is a
Okay.
way. It is here, and I'm just gonna put one thing that because I was the time's sake, I didn't add the codes for the LME4, but I have this here, and I can just post it on the.
Okay, yeah. So I think Christian also covered a little bit of LME4 in the first tutorial. So anyway, so I'm just gonna take one more question, Jales, for you, and then I'm gonna proceed. And then if we have leftover time, we can answer more questions. So there is a
Sounds great.
question from Nazir. How can we get WR VR graph in R using the Heyman approach?
How can we get graph? Well, the graph, I am not. Well, if you have your estimates, you can just plot them in a graph. But I didn't. So my tutorial didn't cover like graph display or how to build your graphs and make your data set look prettier.
Yeah.
But it would be my pleasure. Maybe in the next time, I'll come here and just show something to build graphs.
Okay, yeah, that's cool. So we're gonna hold Jales to what he said. He will be back to show us how to plot some of the SCAs and GCA plots. And yeah, we'll be looking forward to that tutorial. And yeah, so Jales has posted the code for the LMER. Feel free to copy
Yeah.
that code and try it out on your data set. And if you have further questions, remember to copy the JR Biotek. Whenever you email any of the speakers, please copy the JR Biotek in your email so that we can keep track of all the conversations that is going on. And we can keep track of the impact that we're making in the workshops. So thank you so much, Jales. We're happy
Thank you. It
that
was my pleasure.
you shed light on Diallele. At least a lot of people don't have to pay money, a lot of money to have their data analyzed. And sometimes they don't even know if the analysis is correct or not, even after paying such amount of money to analyze their data. So thank you so much. Yeah. So I will move forward with the last part of the workshop, which is to show us how to use another R Shiny package. So how many of you have your R Studio open right now? I want us to do a hands-on data analysis, everybody. Can you show me a reaction? There's a way to show your reaction if you have your R Studio open. Just perfect, awesome. Can you show me just let me know if you have your R Studio installed and open? Perfect. I can see your hands. Alrighty. So I will be sharing my screen right now. Thank you. So today we are going to walk with Mr. Bean. Mr. Bean is a very new R Shiny package released last year. So I've been keeping track of R Shiny. So the package is the package is used for a lot of different types of visualizations, and it uses a lot of different R packages, but you only need to point and click once you import your data. So the first thing I would like you to do is to install packages, DevTools. I would type this in the chat. Where is my chat? All right. So the first thing you need to do is to copy and paste that in your console. And then we need to install Mr. Bean. I want everybody to do this right now. Copy and paste this to line of code. Are we doing good? Is everyone doing okay? Jales, are you able to do this?
Yes, yes, I'm following you. Yeah, I'm following you here.
Okay, so I just want to make sure that everyone is able to install. Abubakar, I can see you. Is everyone able to install this package, Mr. Bean?
One thing that I noticed here, you need to copy and paste individual lines. You can't just copy and paste everything.
Yes, you need to copy the first line first and then copy the second line and run it. So Dawn, are you doing good?
So I'm following you here.
Okay, so I will just wait for a minute while I reshare my screen. Please let me know how you're doing. Okay, so we have a few people who are who have been able to do this. Carol, I don't know, are you able to install your packages yet?
Not yet.
Oh.
I'm not doing it. I'm sorry. I'm sort of doing something else.
Oh, okay.
Sorry.
Well, you need to pay attention here that probably if you have some packages already downloaded, it will ask you to update.
Yeah.
So just press 1 and it will do all of them. You need to press 1 and then press enter. And
yeah. So I would just wait a couple of more minutes. And Daniel seemed to be doing good. Festus is doing good. Abubakar is doing okay.
One last comment here. It will ask you to install from sources. Just press Y and press enter because it is gonna ask you some stuff. So
yeah.
So far so good.
Okay, so once you are done installing the package, if you're not able to catch up, that's okay. I'm just gonna go ahead. Once you install the package, you can just call the package by using the library function. You can do the library and you call Mr. Bean. I already did that. Once you call Mr. Bean, then you need to run the app. Once you start typing run app, the options pop up. You just click run app and click enter. As soon as you click enter, another window will show with the R shiny. So I'm gonna stop sharing the screen right now and share the R shiny screen. Perfect. Can you see my screen now with the Mr. Bean?
Yes.
Okay. Let me just double check with everyone. Okay, so if you're not able to download yet, I will just encourage you to follow what I'm doing. And then after the workshop, you can just continue after the workshop, you can just rewatch the video on Facebook to follow the steps. So Mr. Bean is a recent package developed by these awesome guys. You can see them. Johan, Salvador, Danielle, and Rhoda. They did a very good job because they combined so many softwares and R packages in order to develop a point and click interface for plant breeding data analysis, which is super cool. This Mr. Bean was just recently released, I think last year, and they did a tremendous job doing this. And so I'm just gonna show us how it works. So Nazir says his Mr. Bean is already downloaded. So once you have Mr. Bean downloaded, you need to call it by using the library function and then run the app. Once you run the app, you have an interface like this. So the first thing I would like to show is the data. So on the left-hand side of the Mr. Bean software, if you click on data, you go to upload. This is very cool because you can click here and import your data, your personal data. And if you work with the CG centers and you have the breeding management system where you store all of your data, you can just directly connect the BMS system. You probably have your username and password. You choose your crop, what program, and what trial. You can directly connect to Mr. Bean. And of course, like I said before, you can import data, but I would import data, but I would prefer to use the example data that came with the package. So here is the example data. All right, so one thing you need to know is that your data has to be in the tidy format. What do I mean by that? I mean that every column should contain the variable or the response variable. So these are all response variable, like these are all yield data. This is like 100 seed weight. This is like plant height. This is like days to flowering. So these are all response variables. So you can have maybe yield, plant height, 100 seed weight, stunting or disease resistance, whatever you're working on. And here you have treatment. You also have the blocks, which could also be the replication, but they also have replication here. The row and the column here is gotten from the plot numbers. So this has to do with the arrangement of your field or of your experiment. If you're working in the greenhouse or on the field, there is a way you arrange the pots. So you have to put the number of the row and the column on the field or in the greenhouse. So that is what row and column means in this data set. So once you have your data in the system, the next thing you need to go to the left-hand side, you go to descriptives. So in descriptives, you can actually look at a scattered plot just by one click. So one thing you need to know is R is running in the background while you're doing all of this. So if we want to just take a look at how the rows and the columns are arranged, let's probably group by the blocks, and you click on plot. It's gonna take a little bit though. Yeah, perfect. So once you click on plot, you can see that this is how your field is arranged. Based on the data set that we have, this is how the field is arranged. These are the columns, these are the rows. If you want to see where block 11 is on the field, you can just double click. And you can see this is where block 11 is on the field. If you want to see where block 15 is on the field, maybe something happened to that block, you can just, kind of know where that block is without having to look at your field book. Another descriptive function is the box plot. Once you click this, this settings feature, we'll probably want to see the box plot of the rows and the columns, and maybe we would like to see, let me see, maybe the treatment. So you can visualize the box plot. I know it's going to be a lot of, it's going to take a little bit of time to generate because the data set has so many treatments. So you can easily visualize your data and just kind of see what you have and how everything is going. Okay, this is still loading. So the speed of Mr. Bean has to do with how much CPU you have on your laptop, your processor. It has, it's directly connected to R. So they do have a user interface on the internet, but you know, you cannot know how many people can use it at a time. So it's best to run the app from your R and start the user interface. So the box plot is still running. Okay. So let me see what's going on in the chat. Perfect. First is what, in this case of a missing column. Do you, Festus, do you have a missing column? I don't understand your question. Okay, so apparently the number of treatments are a lot in this data set, so it is delaying. Okay, that's why the plot is so congested because we have so many treatments in this data set. So yeah, let me look at the chat one more time while we wait for that plot to show. So whatever data set you have, you can impute into Mr. Bean. And Mr. Bean will be able to visualize your data set for you. If you don't have like a yield column, if you're missing a column, that's totally fine. The only thing is if you don't have the row and the column, you would not be able to do the spatial analysis, which is the spats. You will not be able to do the spatial analysis. So yeah. So okay. It was a bad idea to click on the treatment. Okay, I'm gonna exit the page. All right, so now that I exit the page, I'm just gonna restart it on R. And Mr. Bean will load back up. Yes, if I do the rep is easier because they're just 2 reps. So yeah, my Mr. Bean is taking so long because I clicked on the treatment. So okay, let's move back to the descriptive. I already showed the scattered plot, so I'm not gonna do that. I'm just gonna click on group variables. I can group them by blocks because I know we have few blocks, but to be safe, I'm gonna do the reps. And yeah. So you can see the arrangement of the field based on row and columns and the 2 replications. And of course, if you're interested in making all of this plot customized to what you want, be sure to watch Alper's tutorial. So this is how you kind of take a look at the arrangement of your data set before you move into analysis. Another option that you would probably want to see is the distribution. The distribution just shows you like the distribution of the variable that you select. So you can select 100 seed weight, plant height, and these 2 flowering. But I'm just gonna select the yield. And here's the bar where you can decrease or increase the intensity of the color. And here is the bandwidth. If you want more bandwidth, you can just raise the arrow without typing any code. The code is running in the background. And of course, we can decide to group the variables based on blocks. I will group the variables based on blocks, and you can see that all the blocks are stacked. This is a really pretty plot. And if you want to view the individual plot, you just check this box. Once you check this box, the individual blocks will be revealed here. And the good thing is you can also see the summary statistics of this, and you can easily download if you want. So this is a histogram of the blocks. You can also do that for like the reps. We have 2 reps. If you want to see the overlap histogram, you can just, by one click, you can see the overlap histogram of the entire trial and to look at the effects of that. So I don't know if we're following, if we have any questions so far. I would like to move into the analysis portion. So, okay, I'm just gonna move into the next one I would like to show is I'm gonna do the trait by trait. Because of our time, I'll only show a few demonstrations today, but feel free to explore, and I'll be happy to show more of this in our next workshop. So now I would like to do the spatial analysis for several traits. So when we say several traits here, this software doesn't analyze them all at once. It analyzes them one at a time. So I will select more than one trait. This is yield. And I would select days to flowering. I will also select 100 seed weight. And I'll just select one more so that we can analyze it pretty quickly. And I will select plant height. So the genotype here would be the line. Your data set can be genotypes or cotton varieties. And the row and the column has to do with the location of that plant on the field or the location of that treatment in the greenhouse or in the lab. Replication would be rep. And if you want to include fixed factors, you can include fixed factors here. And if you have a covariate, you can include a covariate as you want. And then once you do that, you need to run the model. So because we selected 4 different response variables, Mr. Bean will analyze each of the 4 of them one at a time and generate the results for that. So while Mr. Bean is doing that analysis, I think there is a question here. Okay. So you can see that Mr. Bean is feeding the yield data first, and then it will feed the days to flowering, the 100 seed weight, and the plant height. So I encourage you to explore the different functions. You can also do the basic ANOVA, RCBD, CRD, incomplete block design, Latin square, whatever you want, you can do it under the traditional designs. And so it's currently feeding the days to flowering. This is the only function I will show today. Once it's finished feeding and I show you the plots, I think that will be all for this tutorial today. And while we're waiting, I'll just explain the different parts of Mr. Bean. The traditional designs are there. The multi-environment analysis, if you want to see like the biplot, the PCAs, the dendrogram, you can visualize them on Mr. Bean. The only function, however, that I haven't been able to get the license is the ASRAMIL. This software, you need to purchase the license in order to download this function in R. So communication with the lady, one of the persons in the group who developed this Mr. Bean, if we can get a license for ASRAMIL. In our next workshop, we'll probably cover more analysis on that. So perfect. Our analysis is done. So this is for yield, and this is the raw data. And as you can see, the yellow here means more yield. The blue means lower yield. So you can kind of see a field. You can look at the regions of the field where you have low yield. It's probably maybe due to drought or runoff. And this is the feeder data. This is the residuals. This is the feeder spatial trend. And these are the blobs. So this is for trait 1. If you want to visualize trait 2, you can just click on 100 seed weight, and then the plots for 100 seed weight is going to show up. So you can easily see what region of the field you have the highest 100 seed weight. The highest is 70 and the lowest is about 0. Another thing that you might want to visualize is the correlation. So Mr. Bean also shows you the correlation between the traits that you selected before you run the model. So as you can see that these are all not significant. However, plant height has a significant correlation to yield. And you can see that plant height also has a negative significant correlation to 100 seed weight. So these are some of the things that you can see. And of course, down here, you can see the blobs, the prediction and the ranking of the genotypes. If you want to zoom into the genotypes that have the highest 100 seed weight, you just need to click on this zoom reset. You just need to click on this zoom, and you can just like highlight the region that you want to see. And of course, you can see the lines that are the top performers in terms of 100 seed weight. So this you can do for individual traits that you analyze with the model. Another interesting thing that you might want to see is the PCA. So you can see the PCA and the biplots and the individuals as well for every single response variable that you selected in the model. So this is a little bit messy, but this is like a biplot to show the different traits that you selected. We're going to talk more about biplots in another workshop. And here you can see the individuals. In the individual plot, you know that the individuals on the right are the best performers. The ones on the left are the worst performers. So you can also limit the number of data that you can see to 21 to however number that you would like to visualize. So this is a quick way to see the performance of your genotype in terms of 100 seed weight. And these are the bad ones on the left and the good ones on the right. So another thing I would show quickly is the dendrogram. So of course, from the correlation that we saw before, you can see, let me just make sure. Can you see my screen?
Yes.
Okay, my bad. I just wanted to make sure that I...
For just a sec, we lost you, but you're fine.
Oh, holy smokes. Yeah, so I was just like, I hope I'm not just talking to my screen.
No, you're fine.
Okay, so thank you, Jales. With regards to the dendrogram, from the correlation, you realize that the 100 seed weight and the base to flowering had the positive correlation, right? So that's why they're in a similar group. And you can, if you want the lines to be thicker, you can just slide this bar and you can have thicker lines. If you want, like, I prefer the lines to be like 0.5 personally, you know, smaller. If you want the labels to be really big, you can see that. So these are some of the tools we want you guys to use to show your data in the poster competition that will be coming up soon. So we have shown a lot of tools and packages for you to use to show your data in a very nice way. Another thing you would like to probably see is the summary data. So of course, you can see the variance of the genotype, the variance of the heritability, the outliers, the R-squares, the CV, and you can even click to download the table if you want. So you can download the table. The table automatically downloads as a CSV file. So these are some of the tools that we can use to visualize and analyze our data. So there is a lot that Mr. Bean has to offer, but because of our time, I'm only showing the trait by trait analysis. There is also the single site analysis, and there is also the multivariate analysis. And there is also the LME 4, which is an R package that Christian had covered during one of our workshops. And if you click on the model specs, you can see that you can analyze the RCBD, the Latin square alpha lattice, CRD. And if you have other models that is not represented here on balanced designs, you can write the model for yourself here. There is a little tutorial on the left that shows you if you have a fixed variable, this is how you write it. If you have a random variable, this is how you write it. If you have an interaction effect from like your year, your environment, locations, this is how you write it. If you have interactions for fixed effects. So you can actually write the model in here, and it automatically shows you the number of genotypes you have, the number of replications up here. And once you do that, you can run the model, see the effects, and see the summary. So this is a great RShiny package that I wanted to show for us to take time and try it out. So I just want to share my R screen just to show you what is going on. This is my R script. So all of that clicking, you see when I clicked on the LME4, it automatically calls the LME4. So you have to have your R Studio running in the background, because the server, we're not sure if the server can host like so many people working at the same time. So because you have to run the app from R Studio, the capacity and the speed of Mr. Bean will be determined by how fast your laptop or your computer is. So once you're done with running what you want to run, you need to click stop. But I'm not going to stop this now because I don't know if you have any questions. I will reshare my Mr. Bean screen. And yeah. So thank you so much for your attention, people. Do you have questions for me?
There are some suggestions for future workshop here.
All right, Jales, if you don't mind, could you please help me copy out some of these questions?
Yeah, so
yeah, for those of you asking about how to rewatch this video, it was streamed live on our Facebook channel. So let me see if I can get the Facebook page. Just search for JR Biotek and you will be able to find this workshop captured on one of the Facebook videos. And I will see how I can upload that to our YouTube channel as well. So please, we encourage you to like and subscribe our YouTube channel, the JR Biotek YouTube channel, because it gives more visibility to other people to watch and also learn from what we're doing. So don't just watch the videos alone. We encourage you to like, turn the notification bell so that when we have new workshops or training, you can be immediately notified. Someone is finding it difficult to install Mr. Bean. Okay, so for Crystal, for all those having trouble installing Mr. Bean, you can send me an email and I will make sure that Mr. Bean is working on your laptop. So I believe this is a very, yes, Jales?
Can I suggest one thing? Can you just copy and paste all of the codes that you used all at once here at the very end? Because I think if they just copy and paste each line of code, they will be able to download
it. Yeah. So yeah, the thing is, personally, I also had trouble installing Mr. Bean. So I had to run this first. So if you're having trouble installing, you need to run this first. I had to restart R using this code. And then
I think you need to put everyone. I think you're sending a private message.
Okay, so if you're having trouble installing Mr. Bean, you need to restart your R using that line of code first, and then you install the dev tools. There is no R script. This is just a point and click
Mhm.
stuff. Yeah. But R is running in the background. That way you don't have to write any code, but you can generate pretty plots and look at interactions and blobs and, you know, those kind of fancy visualizations for your project. Yeah.
Yeah, so that's
So-
what I was saying. Yes, that's what I was saying. Just copy and paste all at once, and they will probably be able to just follow the steps all at once, because while you were typing, people were like messaging, and maybe we lost some of the path.
Okay, so I will just copy and paste it all at once right now. Thank you, Jales.
My pleasure.
So I hope this was helpful. We're coming real close to the finish line of our workshop today. So someone is asking, is it necessary to remove highly correlated variables before subjecting your data to PCA? I mean, it all depends on, the thing with PCA is most times people use PCAs for the genotypes and not for like the traits. But if you feel that is something you want to visualize, of course, you can even remove outliers, rerun the model by a simple click. And those are some of the things that you can do with Mr. Bean. So yeah. So I will be happy.
Add one thing
yeah.
on that question?
Mhm.
So the PCA is just a tool that generates a biplot that helps you to visualize some trends in your data. So for example, if you want to see the correlations between your traits, you can run a PCA using all the traits that you have measured. So you will see if the traits are close together, they will be correlated.
Yeah.
But for example, if you're using a PCA analysis in a GGE plot, so you're trying to assess the genotype and the genotype by environment information, and you want to see if you can create like mega environments, for example. You can, if you see that the environments are close together, so it means that the G by E effect on those genotypes are similar to that specific cluster of environments. So the PCA is just a way for you to analyze your data, and you get to visualize some trends in your data.
Yeah, so that's great. Thanks, Jales. So someone is raising a concern. He says he wished there would be a session to troubleshoot installation of packages in R. So I think that because a lot of us are having trouble with installation, yes, we can try to cover some installation hiccups and how to, like, you know, get rid of that problem. Yes, we will look into that.
One thing that I will recommend you if you're having a problem installing anything, it might be cheesy, but just copy the problem you're having on. If the error pops up in your R screen, just copy and paste exactly that in your Google, because I guarantee you somebody else has had that problem before and they figured it out. Just copy that problem, put on Google, paste it, and you will find a way to maybe you need to update your R. Maybe your R is not updated, or maybe the package is not in the repository, so you need to use some additional codes. So, but I guarantee you, Google it.
You will find it.
The problem, just copy and paste, you will find it. Yeah. It is incredible how people are working on it and sharing their problems, and it is increasing the network between people that are using R. So it becomes easier for you to find some of the answers you're probably getting. If you're having any problems, you will find a way out.
Yeah, Daniel is saying the same thing. Copy the problem and paste. We'll find solution. Yeah. So yeah, at this point, we're close to 2 hours. So I will hand over the session to the CEO of JR Biotek. We appreciate you giving us the opportunity to share our knowledge with everyone. And I hope that this was useful for you in your breeding program or your research. Thank you.
Thank you. This is amazing. Thank you so much to Catherine. Thank you, Jales. Thank you, Seth and
My pleasure.
Alper. It's been a pleasure, really, to have you all. And it's been really stimulating to learn quite a lot. And I'm really, really proud of Catherine. Like I said, I met her at first years ago at JR Biotek's first training workshop, and that was in Nigeria before she came to the US. And it's amazing to watch her grow so much and to then give back through teaching. And that's amazing. And that's what we are encouraging every one of us here to do. So thank you so very much for your time, the time that you put into to prepare the lecture. It takes a lot of time, especially since you're PhD students and you're doing your own research. There's a lot that is required of us here as well, but then taking that time to give back and share knowledge without charging money is really amazing.
Yeah.
And we thank you for that. And just to tell you all that, the workshops that we give actually, for free, and we're doing it to empower all of you to, do, sorry, I'm wearing a mask and it keeps going back and forth. But this is the new normal, isn't it? We have to work because I'm at work. But really, thank you so much. Please continue to share the knowledge. And if you have any questions, like Catherine said, just put it in the chat box and we will be able to answer it, or send us an email. You can send the speakers an email and copy the JR Biotek Foundation. But at the same time, just be mindful as well how much questions you send. Feel free to send them. If they have time, they'll respond, but don't keep, reminding them every other day that they need to respond to you and you alone. So just be mindful that they're also very busy with their own research and also have to be accountable where they are. So thank you so much once again. Does anybody have any final questions, I think? And another thing that I would say to you is that if there are pressing topics that you need to learn or techniques, it's really unfortunate that because of COVID, we can't really travel now. We should have been in Benin last year, I think, and maybe even this year somewhere else in Africa doing the hands-on workshop, but we can't do that right now. So we do have the virtual workshop that we're planning to do. It might help. So in other words, we're trying to see if we can do some lab demonstration here, and you're watching us from there. You can ask questions live. Some of the basic techniques like DNA extraction, RNA extraction, how to do gene expression analysis, and some of the things that might be useful for you. So we will try. The only problem here too is that we also have restrictions, the number of people allowed into the lab because of COVID. So we'll see. We're exploring possibilities to see if we could do that. And like Catherine said, we want to know more about the type of research that you're doing in the different countries. I think we have more than 20 countries represented here, and that's amazing. But we want to know what kind of research you're doing. So if you're interested, just keep an eye out for information that we will be sharing shortly. And we want to do a virtual sort of research symposium with African researchers, all of you and your colleagues, to better understand your work. We will give an opportunity to about 10 researchers or groups to do a presentation, like a poster presentation with a possible impact of your work. We want to better understand your research and identify needs where we can maybe find possible collaboration if possible, and if funding becomes available. We want to know what the pressing needs are and how they can contribute to food security and improving the lives of other people in the different countries. So keep an eye out for that. We will let you know. And just finally, before we go, is the survey. Catherine, can you share the survey link on the chat?
So please click on this Google document and please fill out the survey for us. We want to get a count of, how much, what is the area of your research, and we're intending to do some sequencing project, and we want to have like an overview of the kind of crops that are in Africa and do not have a sequenced genome. So please access the form in the chat and please fill it out. We will be grateful. And so for those asking about GWAS analysis in R, personally, I did that tutorial. So what I would do is I will get that video on our YouTube channel and label it GWAS analysis with R, because it's on a separate YouTube channel. That's probably why you cannot find it. But there is an analysis using R for GWAS. And I would also have someone else cover QTL analysis using R. So, in our next workshop, we're going to cover, based on your responses, we will cover some of the areas that you need clarity on. And we'll be able to provide that information for you in our next workshop. So
Yes.
please feel free, to reach out to us and let us know. If you need more clarity, more insight, we'll be happy to provide that as time permits. The link is not working.
Okay.
Well, it is working here. I don't know.
Okay.
Okay, I think one of the things we could do maybe is to send out a survey at some point just to know what topics and what sort of areas the majority of our researchers in Africa are interested in.
I think the link needs permission. Let me see if I can edit
it-
that permission aspect of it. Oh, okay. You can try it now. It should work. Yeah, I just removed the permission. Yeah, you can try the same link now. It should work just fine.
Okay, so we need to bring this to a close.
Okay.
Yeah, so once again, thank you so very much. And again, Catherine, have you sent the link to the survey? Can you put it on the chat here? So if you could just fill out the survey. And again, like Catherine said, we're looking to identify African research groups and researchers that are working with African indigenous crops or orphan crops that the genomes have not been sequenced. We're looking to see if we can raise some funding to set up a partnership to sequence some of these crops, the genome of these crops, and assemble them, apply bioinformatics with some help here in the UK and in the US with our African researchers. And that will be to provide the resources that, most of the researchers who are working with these crops with unknown, nobody knows anything really much about their genomes can. So, potential crops that can help to address hunger, and malnutrition in our different countries in Africa would be ideal. So please do fill out the survey to tell us the crops that you're working with, if they're orphan crops, if the genomes have been sequenced and what you're doing, and that will help us to then make a decision on how we would go about finding the right sort of research groups or researchers, some of you, to work with, and how we aim to address this issue, which I think will be an excellent way to sort of, contribute to addressing some of the pressing questions regarding food insecurity as well. So thank you so very much to our speakers. I wish it was in person to give you a clap, but I'll clap on behalf of everybody else. You all are amazing. And of course, you'll come back soon to do a little bit more teaching. And thank you to our African researchers for always responding. Please do teach with the materials, do share the knowledge with others as well. And we're here for you. So thanks again. And please stay safe out there.
Yes,
thanks.
please. Yeah, thank you.
Yeah.
And please watch
you,
out
everybody.
watch out for our poster competition announcement. And
Catherine, have you sent the link still? I haven't seen it in the chat.
Oh, it's
for
there.
the survey. Feel free to unmute yourself to say goodbye. You can now. You can unmute yourselves and say goodbye.
Yeah, it was a pleasure, guys. Thank you very much for your attention.
Thank you,
and
Jales.
I hope you guys learned something this morning. And my email is displayed on the lecture. So feel free to shoot me a message. It'll be my pleasure. It was an honor to be here. Thank you very much for the opportunity.
Thank you. Thank you so much. It was very, very, very interesting. I did learn as well. I did take notes here on the side. And sometimes I do go back to the videos to watch if I have some questions, right? So, I mean, it's not just for the African researchers that this is actually really, a lot of people in Europe and America also use the lectures that we post on Facebook.
Yes.
And
That's
they
great.
do. I've used
That's
the
great.
CRISPR one. That I can't remember his name, from Texas A&M as well. I did have a few points there that I did watch the video trying to design CRISPR knockout in wheat, even though I did it in rice. But there were some really important information that he shared that I wasn't aware when I did my first design, even though it worked somehow. But those things are useful. So thank you so much for sharing that knowledge. We appreciate it.
All right.
Okay. All right. Thank you.
Thank you very much, everyone.
Thank you.
Thank
Thank
you.
you very much.
Yeah,
Good
Good
night.
you, Catherine. Thank you,
Guys,
Carol.
have a-
Thank you.
Thank you.
Please
Thank
do
you
fill out the survey,
very
okay?
much.
We really
Alright.
appreciate.
Yeah, we are.
Thank you so much.
Thank you.
Thank you.
Bye from Kenya, South Africa, Nigeria. For some reason, Nigeria.
Yes.
I can't hear you all.
We have
We're
a few
all-
people.
We
Oh,
are
okay, they
here.
are.
Nigeria, we are so many.
Oh, of course, if Nigeria is not there.
Bye bye.
Oh, okay.